Mircod Biotech POC is an automated, multifunctional, closed-loop system for manufacturing genetically modified cell products. The system procedure for cell growth is both reproducible and scalable. While discoveries using genome-based technologies have accelerated, we drive change to beat oncological and rare orphan diseases and support innovations in regenerative medicine. The system optimizes cell transduction and culture processes with customizable, single-use disposable cartridges to match the requirements of the particular cell type. As a result, significantly greater cell viability than traditional manufacturing process, higher transduction efficiency, and lower variations of cell subset distribution. The system incorporates the latest developments in bioprocess control automation with innovative features implemented by our R&D and biotech teams to maintain a consistent and predictable environment, automating critical processes such as transduction, cell feeding, exchanging gases, QC, and cell harvesting. Point of Care is a state-of-the-art biotechnological modular system that consists of an extensive range of interconnected components that can also act as standalone biotech research and production instruments and tools, making it commercially viable and widely applicable. The remote control with web-based extended functionality and wireless detachable smart monitor provides an intuitive user experience, reduces operator errors, and improves response time to events and incidents. Custom adaptation and configuration of a single-use cartridge and predefined, clinically verified, combination of cell culture protocols, flexibility in equipment, and process automation creates a significant advantage over the old-school systems and processes. Depending on the desired therapy needs or disease-specific characteristics and the patient's individual profile, scientists and doctors can deploy a range of production scenarios from preclinical research all the way through to current good manufacturing practices, cGMP, for commercial production. A rapidly emerging immunotherapy approach called adoptive cell transfer, ACT, begins by collecting and using the patient's own immune cells to treat their cancer. The process requires drawing blood from patients and separating out T cells. Next, using a disarmed virus, the T cells are genetically engineered to produce chimeric antigen receptors, or CARs, on their surface. Within a sterile environment, the system automatically separates T cells. The steps for cell preparation include automatic filtration, washing, and concentration procedures. The cells then go through the process of transduction, a technique by which foreign DNA is introduced into a cell by a virus or viral vector. Finally, once the collected T cells have been engineered to produce the antigen-specific CAR, they are expanded into hundreds of millions of cells in the specifically designed reactors. During all processes mentioned above, the system monitors the effectiveness of cell growth by tracking all necessary vital biomechanical markers of the modified immune cells. Flexible and custom-created QC procedures help researchers to perform multiple operations in different places simultaneously. The POC maintains predefined temperature, nutrient supply, and manages waste disposal. It also controls the preparation of the medium for cell culture and performs a cytomorphological study. 2 to 3 weeks later, according to the QC results, an automatic harvesting procedure is initiated and the CAR-T is collected in a specified amount and divided into doses to be sequentially administered to the patient. A predefined part from the overall yield will be separated and stored in liquid nitrogen cryobank. These cells can be used later according to treatment requirements. If all goes as planned, the engineered cells, once injected, further multiply in the patient's body, and with guidance from their engineered receptor, recognize and kill cancer cells that harbor the antigen on their surfaces. Unlike traditional cancer drugs, such as small molecules and antibodies, that rely on passive targeting, T-cell-based therapeutic agents can actively home in on the disease sites, sense the microenvironment, and integrate multiple inputs to mount a complex and dynamic immune response. Once the job is done, in order to control severe CAR-T cell toxicity, a special code activates a mechanism for rapid ablation of the transplanted cells through a suicide switch. Controlled remotely, the system is fully automated and cGMP compliant. The process of cell activation and cultivation can be adjusted as needed. All devices support telemetry control, share a common data cloud, and are interconnected in a global network. All successfully completed cycles are recorded and flagged by the system. If needed, they can be easily reproduced on any other device. Mircod Biotech POC is expected to reduce labor costs by 70% and lower the price of these specific therapies from the $300,000 to $500,000 range to the $60,000 to $80,000 range.